Good morning to everyone. This is Thursday morning. It's 10 o'clock. Means it's Enjoy Science, and also means it's another wonderful colleague and speaker, Dr. Sam Hanash. And thank you very much, Dr. Hanash, for your time to share with us wonderful science. So I want again to thanks to you who are attending the webinar, because without you, the webinar cannot happen. I want to thank, of course, to Giulio Draeta, because this is in combination with the Chief Scientific Officer, and of course to the Translational Molecular Pathology, Dr. Ignacio Vistuba, and also to thank very much to Emily, because she is the one who is organizing, keeping this organized, and to James, who is recording the session. With this, let's be happy to be so lucky to have here Dr. Samir Hanash. All of us in MD Anderson, we know Dr. Hanash. Why? Because he's a wonderful colleague, all the time ready to collaborate with everybody. Probably you know very well, he has an MD from American University of Beirut in Lebanon, then a PhD from University of Michigan, Ann Arbor. And then he basically worked up in science, becoming, to be fair, one of the world leaders of proteomics. We were very happy to bring him here from Fred Hutchinson. And in fact, Dr. Hanash set up in a great way the true proteomics work in our institution, which was very helpful and in big need. He is a director of McCombs Institute for Early Cancer Detection, also a co-director for the Department of Global Cancer Early Detection. And what I want to say is not what are his titles, we know very well. I want to say that he's extraordinarily important for our mission. His publications are extremely high level, and I can tell you, working already with Dr. Hanash for several years, he organized one of the most, if not the most, formidable database of proteomics in the world. If you have in your head any idea, any protein, any peptide, I can guarantee you Dr. Hanash has tons and tons of data. Why? Because he's extraordinarily organized. He's keeping everything in databases, and really his lab and his data, his work, it's a wonderful resource for the institution. With this, thank you very much, Dr. Hanash, with being with us, and we are all looking forward for our talk. Thank you very much.
Well, thank you, George, for this introduction, and thank you for the opportunity to present our work related to liquid biopsy and cancer screening and early detection. It's a beautiful day in Houston. I hope you're having a good time wherever you are joining us, and I appreciate your joining us. In terms of disclosures, I have filed multiple IPs pertaining to biomarkers, and I've done a little bit of consulting for different companies. And so where are we at the present time? So despite progress, cancer remains a largely unsolved clinical problem with high mortality. And in order to reduce mortality related to lung cancer, we have to move on multiple fronts. We need to identify who is at risk of developing cancer, so we could implement preventive interventions, like, for example, vaccines, which we know is working for COVID. There is no reason why vaccines would not work for cancer. This could be one way of intervening even before cancer is detected. And if we cannot prevent it, then having means to detect cancer early obviously would contribute to reducing mortality. Beyond that, we need to personalize treatment and be able to monitor disease as it progresses. So we have to move on all those fronts to reduce mortality. And I'm going to cover the first part here, which is risk assessment and early detection. And so back some 2 decades or more ago, the NCI organized a meeting to assess what we can do in relation to early detection. And so it was clear at that time that we have developed a deep understanding of cancer causation, genetic drivers, the host response, and that we had available game-changing technological advances like genomics, proteomics, metabolomics, immunomics, and ways to assess different markers in a multiplex type of fashion. And that it was felt creates an opportunity for implementing strategies for cancer early detection that would be at a modest cost through the application of biomarkers. So that kind of workshop at that time led on the part of the NCI to develop the Early Detection Research Network to stimulate collaborative discovery, development, and validation of biomarkers for cancer risk assessment and for early detection. And a number of us at MD Anderson have been involved in the Early Detection Research Network ever since it was launched quite some time ago. Now, at that time, the field of early detection was sort of kind of perhaps a way of describing it was that it was a little bit on the obscure side. It wasn't clear whether this is going to work or not work. And it was sort of, the field was limited to some aficionados who had a conviction that this will work. Now, over the past 2 decades, there has been an incredible heightening of awareness of the needs and the opportunities for early detection. And there's been flooding of social media about the public wanting to see that field move in a much stronger fashion than it has been. And so, like, for example, one of our fellow Texans on his blog had this message that he sent some time ago, that a big failing of medicine is that we wait till we are sick to have our blood tested and compare the results to comparable demographics. And he got comments from other people saying, yeah, a big failing of society equals reactive instead of being proactive in relation to disease. Another one said, we need to be more proactive versus reactive. We have a louder voice that can help. At the same time, the conclusion of all of this was that the bottom line, blood test results are your health resume. They offer snapshots of your health at the molecular level, and they provide a spectacular early warning system to identify illnesses prior to symptoms. This is not coming from a scientific or from a public health person. This was coming from, well, if you can call Mark Cuban an average person, somebody with a little bit of insight about the need for much better blood testing so we could catch disease early. Now, he's not the only one. And here's another editorial in the New York Times. Women need better medical tests, not more treatment. Instead of mammography, which finds many lesions that will never spread, women need tests that find disease that truly needs to be treated. It's clear that there's a recognition that the US national expenditure for false positive mammograms and breast cancer overdiagnosis, $4 billion a year. Something should be done about this. Another post by Scott Gottlieb, who used to be the FDA head, overwhelming data show early detection of cancer is key to long-term survival. So the public is sort of kind of reacting and being flooded with messages about the need for early detection, better ways to assess disease. And if we could catch disease early by way of blood tests, that would be good. So that's kind of like the message that was out there, not 20 years ago, but in the past recent few years. And back now, when was this? In 2016, when at the JP Morgan Healthcare Conference in San Francisco, CNBC had this write-up about a revolutionary blood test that can detect cancer. Shock and awe. We went from a few scientists and academics wanting to kind of work on the field of early detection, now big investors wanting to see there's an opportunity, $20 billion out there related to cancer being detected early. Let's move in on it. And what happened? It created a little bit of a gold rush with many, many investment groups putting in hundreds of millions of dollars toward developing blood-based tests for early detection. And who are some of them? And here's just like a little list of some of the companies out there that have gotten hundreds of millions of dollars to do this kind of work. Everybody, it seems by now, knows about GRAIL and their Gallery test for multi-cancer early detection. They can detect 50 cancers and they can save lives with that approach. Thrive has licensed the cancer seq test that was developed at Hopkins that I made some contribution to. Then they were later bought by Exact Sciences on account of the promise of a blood test for multi-cancer early detection. Guardant is basically about to launch a colon cancer screening test, and I'm sure they are interested in working on other cancers. Yet another company, Freenome, has amassed hundreds of millions of dollars to develop liquid biopsy tests for cancer early screening, and I got a kick out of their messaging about them being pioneering early cancer detection with proteomics, something that a number of groups are working on for well over a decade. And the most recent addition is our own Steve Hahn, who used to be at MD Anderson, was the FDA commissioner, and after he left the FDA, now he's the CEO for a new company that has been launched, Harbinger Health, to pioneer the detection of early cancer by a blood test by harnessing artificial intelligence and the biology of cancer's origin. So we went from this being a little bit of an academic activity whereby the science moves very slowly, incrementally, to now literally billions of dollars going into this field. And so what's happening at the present time? So a lot of people, especially at the company level, are very optimistic about their view of liquid biopsy for early detection. And they basically believe this is going to be a very winning strategy. On the other hand, we have people who are pessimistic. They have a view about liquid biopsy that this is not going to work to be able to look at 50 cancers and be able to catch them very early to be effective, that there could be a crash along the way. So we have to be very careful. So you have those two camps, the optimistic people and the pessimistic people. And so, and again, there's a lot of debate going on now. And here's one article, Liquid Biopsy, Misplaced Faith in Early Cancer Detection. In particular, companies engaged in using a single blood test to detect multiple cancers, so-called liquid biopsies, are attracting a lot of investors, billions of dollars. Both the companies and those funding them are banking on the public space in the claim that early cancer detection always saves lives. So shedding some doubt about all of the hype around early detection. Another here, scientist, a colleague, and maybe a friend of mine at the University of Toronto has been showering the literature with papers about doubt about liquid biopsy working as being promised. The one paper after the other, and here's one. Circulating tumor DNA, ctDNA, is not a good proxy for liquid biopsy of tumor tissues for early detection. And his estimate is based on the fact that when a tumor is the size of a few millimeters, then in order to shed DNA into the blood, you need to have very large volumes of blood to detect a single molecule of DNA. Therefore, this thing is not going to work. Well, okay, so at my end, we had this opinion piece in Cancer Cell with Sudhir Srivastava. Together, we wrote a piece about pan-cancer early detection. Is it hype or hope? And so we sort of kind of presented both ends of the spectrum and felt that somewhere in between is a judicious sort of position to take. And so what are the issues? What are the challenges with multi-cancer early detection? Well, first, if you're going to detect 50 cancers at the same time, then you better have a very, very, very high sensitivity, specificity, sorry. So you need that high specificity because a 1% false positive and you're looking at 50 cancers, that's going to be extremely detrimental. So what is the problem with having high specificity. The problem with high specificity is that almost by necessity, it will reduce sensitivity. So you're going to be missing out a lot of cases. The other thing is that when you look at what some of the companies are doing, like for example, Grail, when they started out, they thought that finding mutations in the blood is going to be the way to catch all of those cancers. Then later became clear, well, that's not working. They shifted from looking for mutated DNA to looking at methylated DNA because there are so many more sites, millions of sites of methylated DNA. It remains to be determined whether a single marker type will be sufficient to achieve adequate performance for detecting cancers. There's also the issue with multi-cancer early detection pertaining to the test being positive, but you have no idea what the cancer is. How are you going to deal with that? And so those are some of the issues. The counter argument has been that, well, even if the sensitivity is low, any cancers that you catch is better not catching any cancer, and you will save lives that way. So at the same time, basically, when you read all the different opinion pieces in favor of multi-cancer early detection, what comes across is that no one considers MCED, multi-cancer early detection, as a substitute for screening with current modalities that are available. Yes, go ahead with the screening that's available with lung, breast, colon, and cervix, and our test is not going to substitute for those modalities. So what does that mean? So the position that we are taking is that the multi-cancer early detection test, if it's not going to substitute for screening for the cancer for which there is screening, at least it should empower screening with those types of tests for lung, breast, colon, and cervix. But for those cancers, there is no screening for them at the present time. That's a different situation. So you cannot have like the same requirement for performance for tests for cancers for which there is screening compared to cancers for which there is no screening. And so we have to individualize the performance of the blood test, whether it's a single blood test for looking at only one cancer or a blood test looking for multiple cancers. We have to individualize the performance based on cancer type by cancer type. So if there's no screening available for a particular rare cancer, let's say a sarcoma, for example, and your test is intended to detect sarcoma, then it better have very, very high specificity. And in that case, you can make the argument that catching 10% of sarcomas is better than not catching any early. Yeah, you can make that argument, but for cancers for which screening is available, you want the blood test to empower screening. What does that mean? You want the test to bring to screening subjects that otherwise would not have been screened on the one hand, and two, reduce the false positivity associated with the screening modality itself. And so the screening that we have today for breast, we have mammography, colon, colonoscopy, lung, low-dose CTs, and what have you. And so the problem with those tests is that they have limited sensitivity and specificity, limited adherence and cost, and they have potential adverse effects. So the approach that we are taking is, would a blood test improve on the performance of the modalities that we have today for those types of cancers? And if a blood test can do that, that's a tremendous, tremendous contribution. And so that's exactly what we have been working on, namely going after a relatively small but very common cancers with substantial mortality, namely lung, breast, colon, pancreas, ovarian, gastric, where we want to develop a test, a blood test that determines a subject's risk for those cancers. And based on that risk, then they would be followed with a screening imaging, usually modality. For prostate cancer, we don't need to detect more prostate cancers. What we need is to develop predictive markers, which prostate cancers are going to progress and which ones are not. And so those are the cancers that we were working on for those specific applications. Now, if you sit down with a pencil and a piece of paper and ask, what could there be in the blood that would tell us about somebody harboring a cancer at an early stage, or even kind of pre-cancerous lesions that we can monitor for? Well, you have a whole laundry list of things in the blood. So you could look at nucleic acid, mutated DNA, methylated DNA, non-coding RNA, or coding RNA. You could look at proteins, metabolites, cytokines, chemokines, autoantibodies, exosomes, microparticles, circulating tumor cells, a whole laundry list. And so how do we deal with that? So at our end, we are trying as much as possible to leave no stone unturned and look at every which modality and see what it can contribute. And so this is our approach to discovery. Try as many of the platforms as available that you have access to. At the same time, something that distinguishes what we are doing from practically everybody else is that most of the studies that you read about, whether they're coming from companies or coming from other academics, the discovery is usually based on looking at blood from subjects already diagnosed with cancer. It could be advanced stage or even early stage, but those subjects have already been diagnosed with cancer, and trying to come up with a signature that may or may not work at stages when subjects are totally asymptomatic, they're running around, they don't know that they have cancer. At our end, we're doing something different. We're doing the discovery work in preclinical samples, meaning that at the time the blood was collected, the subject did not know that 6 months later, a year later, 2 years later, 5 years later, they are going to be diagnosed with that cancer. And that's how we do our discovery. And so we work with cohorts of subjects where blood was collected and later they were diagnosed with cancer. And we would ask, okay, the blood from that subject that later was diagnosed with lung cancer, do we see anything in that blood that distinguishes that subject from somebody who was followed the same way, had the same subject characteristics, but did not develop the cancer? At the same time, what we have done is to integrate our findings from humans with findings from mouse models of cancer and with what we see in tumor tissue and tumor cell lines. And the nice thing about mouse models is that you can turn on the cancer-causing gene and start sampling the blood and sampling the tissue. And under sort of kind of uniform standard conditions where there are not so many variables that you encounter, obviously, with humans, with a lot of heterogeneity with what they do and all kinds of factors. And so the mouse is kind of good for that biological angle. And we're not relying on the mouse alone, but we are kind of integrating data from mice. We used to be part of the Mouse Models of Cancer Consortium when it was active. And I had access to a lot of mouse models, and we'd integrate data from mouse models with data from cohorts. And once we identify a marker, we would do the validation of that marker also in preclinical samples, as opposed to just newly diagnosed with cancer. And so we had access to a lot of mouse models of cancer with our collaborators, and they are listed here. Mouse models of lung cancer, multiple cancer, models of lung cancer, multiple models of breast cancer, pancreas, colon, ovarian, prostate, models of inflammation as kind of like non-specific to account for non-specific findings. And we did a lot of profiling of those mouse models early on during tumor development. And so I don't have time, obviously, to cover all of the work that we've done for every which cancer, but I'm going to focus primarily on developing a blood test for lung cancer screening, and maybe a little bit, if there's time left, to talk about a couple of other cancer initiatives that we have going on. So most of the rest of the talk is going to be about developing a blood test for lung cancer screening. And so what do we know about lung cancer screening? Well, we know now for more than 10 years that subjects who are at risk of developing lung cancer will benefit from CT-based screening. And this was the famous National Lung Cancer Screening Trial that was published in 2011 that shows that mortality can be reduced among smokers who meet certain criteria if low-dose CT is implemented. Now, this is great. It was really a landmark study, but we have recognized that there are challenges with CT-based screening. First, there's a high percentage of false positive where something is seen on the CT that turned out not to be cancer. And the other thing is that if all smokers and former smokers do 3 CT scans per year per NLST protocol, the healthcare system is going to be totally overwhelmed. That was the view. And so where would a blood test fit in? Well, first of all, not every subject who is eligible for screening actually is going through screening. Back at the time when we launched this study, then the USPSTF criteria were 30-pack year smoking history and or subjects who are currently smoke or who quit within the past 15 years. Now, they reduced that requirement to 20-pack years. But then for subjects who are eligible, but then are not going through screening, who are not even eligible, which represent a vast majority of subjects at risk of being diagnosed with cancer, that ultimately will be diagnosed with cancer, what can we do for those subjects? And we felt that a blood test that determines somebody's risk would be very impactful to reduce mortality because it would lead to a much, much better and more efficient implementation of CT-based screening. And so the first kind of collaborative group on this theme that I led was funded by the Department of Defense now some 10 years ago. And again, we wanted to develop the best marker panel possible. And we had teams working on mutated DNA, methylated DNA, microRNA, proteomics, metabolomics, cytokines, chemokines, autoantibodies. And we had a group at Sage Bionetworks at that time that was in charge of integrating all of this data that we had generated. And so I mentioned earlier that we are working with cohorts of subjects before they were diagnosed with cancer. And I feel that we have been very privileged to have had access to those cohorts. So for the lung cancer biomarker work that we did, the sort of the major effort early on was on working with the carotene and retinol efficacy trial, 16,000 subjects who were largely smokers who were at risk for lung cancer, and quite a lot of them ended up developing lung cancer. We had access to their blood. We have used that cohort for pancreas also. We have worked with the Physicians Health Study on lung cancer biomarker validation. We have worked with the 160,000 Women Health Initiative cohort, WHI, to discover markers for lung, breast, and pancreas. We worked with the Cardiovascular Health Study, 2 cohorts, 500,000 set of subjects in Europe. We have worked more recently with the NCI PLCO cohort. I'll talk a little bit about that, a colonoscopy cohort, and we are now working with the Canary prostate cohort, active surveillance of subjects who are diagnosed with prostate cancer to validate markers that we have. So those are well over 1 million subject cohorts that we had available to do our discovery and initial validation work. Now, those are obviously cohorts outside of MD Anderson, but we also have access to cohorts at MD Anderson. I'll talk a little bit at the end about the LEAP and the MERIT cohorts for lung cancer screening and mammography. We also have a breast cancer high-risk cohort, BCIS and germline mutation, a Mexican-American cohort, a normal risk ovarian cancer screening cohort, and we have our own past prostate active surveillance cohort that we have access to. Now, having access to a lot of blood samples is one thing, but quality is extremely important. So we have been at times reluctant to be using this or that cohort because we did not have good, sufficient information about the process by which samples were collected, quality of the samples. And so we want to make sure that we're dealing with quality material. This is just like an illustration of how blood collected could look so different with hemolysis and other things that mess up the sample, that it makes it unreliable to use either for discovery or for validation. So quality is very important. And this came into kind of a real focus with the PLCO cohort when this analysis was published by the leaders of the PLCO cohort, namely that a lab detective work identified a mishandling problem in sample aliquoting. And they realized that with some of the samples, if you had drawn your little volume to do your assay of CA125 from the bottom of the tube, compared to drawing that small aliquot from the top of the tube, you got different values. So quality control of samples in doing this kind of work is extremely, extremely important. So our first landmark study, going back to integrating mouse models with studies using human samples, came out in Cancer Cell, made the cover. And at that time, we were using mass spectrometry to profile blood to see if we could find differences between different mouse models. It was a simple question that we were asking. We have models of lung, breast, colon, ovarian, what have you. If we draw blood and do mass spectrometry analysis on that blood, can we distinguish lung from breast from colon? And the answer was yes, it was pretty readily using unsupervised hierarchical clustering. We could distinguish between those cancer types. What was interesting is that there was a signature for lung cancer among the 4 different mouse models of lung cancer, as you could see on the bottom. So some proteins were up, those are the ones in red, and some proteins were down. And so we wondered, like, what is it that connects those proteins in circulation that were up in the different models of lung cancer? So we went back to gene expression data and did some analysis to see what kind of correlates with this sort of a signature for lung cancer. To make a long story short, it turned out that the signature is related to expression of the proto-onca gene NKX2.1, which has been known to be associated with lung cancer. So NKX2.1 is a lineage survival oncogene in lung adenocarcinoma associated with early stage disease and with good outcome. And then when its expression goes down, then you're dealing with high grade, poorly differentiated tumors. And obviously we're looking at very early stages. So we did have an association, but it wasn't just statistics that told us, oh, here's the marker for lung cancer. We have biology behind the association, namely an NKX2.1 signature. Now, what were the proteins in that signature? One of the proteins was surfactant B. And we were wondering, why would surfactant B, which is obviously a physiologic factor related to the health of our lungs, associated with lung cancer? And so we looked more closely at the details of what we identified about surfactant B. And it turned out that, in fact, what we were picking up in early stages of lung cancer development was not surfactant B, it was pro-surfactant B, the immature form of surfactant B is overexpressed with the development of cancer. And so when we made a comparison of the performance of mature surfactant B, which you see on the right here, the black curve, and compare the performance with the immature form of surfactant protein B in the red, then pro-surfactant B was much better performance. And so given those findings, and there was no antibody available against pro-surfactant B, then we had an antibody assay developed against pro-surfactant B. And so once we had the assay, we went back to yet another cohort, the Canadian screening cohort, lung cancer screening cohort, and had access to blinded samples. We just wanted to see the performance of pro-surfactant B and compared that performance to what we know about risk associated with smoking and other factors. And so what you see in red here is performance based on a risk model for lung cancer due to subject characteristics. And when you compare that with the performance of prosurfactant B, you see an increment in performance. So that led to this paper with our colleagues at University of British Columbia in JCO 2013. That was accompanied with an editorial about the promise of blood-based testing for lung cancer. And so this was a proof in a blinded fashion of the merits of prosurfactant B for lung cancer risk assessment. So I don't expect you to read every number here. The point of this slide here is to tell you that when we looked in greater detail at the data for prosurfactant B in the pan-Canadian screening cohort, every which way we looked at the data, those that were later diagnosed with advanced stage versus early stage, male versus female, squamous versus adeno versus small cell, the p-value between cases and control was super high every which way we looked at data, 0.0000 or 0.001 type of thing. And so that was very encouraging for us that prosurfactant B is a very robust marker to assess risk of lung cancer. What was also totally astounding was that in the pan-Canadian cohort, there were subjects that at the time of screening had a negative CT, at the time of screening had a positive CT that did not turn out to be lung cancer, or that turned out to be cancer. And they had subjects that had a negative CT that a year later were diagnosed with lung cancer. And we asked, well, how did prosurfactant B perform in those groups? What was astounding was the highest performance that we saw was in that group that had a negative CT, but a year later was diagnosed with lung cancer. So a year before CT was positive, the prosurfactant B marker was highly expressed or detectable in the blood. So that again reinforced that we have something going on there that is associated with risk of lung cancer. So that led to when I moved to MD Anderson to launching, as part of the Moonshot program, a lung cancer early detection project. And so the goal of the project was to identify subjects at risk for lung cancer based on their biomarker profile and subject characteristics. For those subjects that we are going to screen, provide access to smoking cessation program, and basically improve the reliability of CT screening for lung cancer early detection. So we had markers of our own at that time, but obviously we did not want to blind ourselves to whatever other people have also discovered that look promising. And so we reviewed the entire literature about biomarkers for lung cancer at that time. And there were 10,772 papers that basically mentioned something about a potential biomarker for catching lung cancer in early stage. So we systematically went through all of this, at times just reading the title, at times reading the abstract, and at times going through the paper. And we decided that we're going through pulling out relevant markers to do pre-validation and ultimately leading validation, going through sequential validation to come up with the best marker possible. So we had access to pre-diagnostic samples from well over 1 million subject cohorts that we're going to do the triage of markers on. And then we're going to rely on the pan-Canadian, the Pittsburgh screening cohort to come up with a risk model that we would validate in the NCI PLCO cohort, leading ultimately to a prospective validation study. And so in this approach of trying to triage biomarkers, we had to carefully select anchor markers as panel members, with prosurfactant B being our number one anchor marker. But also we had to look into, well, can we assay this other potential marker in a reproducible fashion? Can we come up with robust assays? And can we combine anchor markers for the best performing panel possible? And so looking, for example, at literature for microRNAs, there were a lot of papers that talked about microRNA for cancer early detection. And the challenge was, in some paper, they identified X number of microRNAs that looked very promising. The other paper did not identify any of those microRNAs. What to do, what to do, what to do. So we ended up with a panel of 81 microRNAs that seemed to be the most plausible that we went through a validation study on. Okay, that was microRNA. What about proteins? There were umpteen proteins out there that were either being used as part of workup of patients with lung cancer, or that were described as being promising. Okay, we assembled all of those promising protein markers. And so in the work that we were doing, another marker surfaced, which was a metabolite, and that was diacetylspermine as being elevated before a diagnosis of lung cancer. Since prosurfactant B was already our anchor marker, we asked, does diacetylspermine add to the performance of prosurfactant B? And the answer was yes. So this became a very reasonable marker to want to add to our panel, and that led to another JCO publication in 2015. Now, so we were working with so many different markers, and at the end, at some point in time, then we felt that prosurfactant B plus this metabolite diacetylspermine, if we were to add 3 microRNAs, 2 additional proteins that look promising, we can improve performance. But then at that time, we were talking to the Roches, the Abbotts, and others who are obviously, who have very leading platforms in the clinic for biomarker testing. And the answer was, Sam, there's no way we're going to implement some kind of test for lung cancer that has a bunch of different things, microRNA, metabolite, proteins. No, 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 this is not going to work. Okay, well, what would work would be, well, okay, if you want more than one marker, at least have the markers to be of the same kind. And so at that time, we got to that point, we thought, okay, let's assemble the best promising protein markers that we had. And this was basically a combination of 4 markers that included prosurfactant B that we developed using the CARET cohort, and then we locked down the combination using the CARET cohort. And then we reached out to our colleagues in Europe at the International Agency for Research on Cancer. Actually, they reached out to us saying, hey, we have 2 cohorts with 500,000 subjects and quite a few developed lung cancer. Would you like to validate your marker panel with our samples? And obviously the answer was, yeah, we would like to do that. Send us the sample blinded to do validation with pre-diagnostic samples and see how our 4-marker panel will perform. And so, and that came out in JAMA Oncology in 2018. And so to make a long story short, then when you compare the screening criteria that were available at that time in terms of their performance in assessing risk of lung cancer, the 4-marker panel plus subject characteristics improved the sensitivity at the same specificity and improved the specificity at the same sensitivity. So we're very excited about that. And so we had applied to the NCI to do as a U01 grant with the rationale that we have data that support the use of a 4-marker panel for screening. And we would like to use the NCI PLCO cohort to do further validation. The grant was approved and we got the samples to go with it. And we had access to 10,605 samples to do the validation work. And those included lung adeno, squamous, small cell, and we put perhaps a little bit more emphasis on the subjects that were diagnosed within 1 year, but we also did validation on subjects that were diagnosed within 5 years. And to make a long story short, when we combined a risk model based on smoking and other characteristics, the so-called PLCO 2012 risk model with our 4-marker panel, we got very good performance on non-small cell lung cancer, and that included adenosquamous as well as small cell lung cancer. with AUCs of 0.85, 0.84, 0.88, 0.85, very similar across all the different subtypes. Now, at the present time, the USP-STF criteria have changed, so they have lowered smoking requirement to 20 pack years instead of 30 pack years. And what we have on this slide is the performance of our marker panel compared to the current USP-STF criteria. So you have a black cross here, I hope you can see it, and a gray cross. The black cross represents the old criteria, which had greater sensitivity, limited specificity, and the less sensitivity, but greater specificity, and the new criteria, which had more sensitivity and less specificity. But when we compare the performance of our marker panel compared to the current criteria, at the same specificity, again, we get better sensitivity, and at the same sensitivity, we get better specificity with the current criteria. To make a long story short, the 4-marker panel plus the risk model based on subject characteristic had improved sensitivity, 88.4% versus 78.5% for the current screening criteria, and improved specificity of 56% compared to 49%. The upshot, if this test were to be used for smokers who don't even meet the eligibility criteria, 10-pack year smokers, the marker panel would result in 9.2% more lung cancer cases being detected and 13.7% fewer non-cases that did not need to be referred to screening that otherwise would be referred for screening. So we have done other types of markers, and again, we leave no stone unturned. And here's a comparison looking at the performance of ctDNA for mutations, the work that we were doing with Hopkins, Bert Vogelstein there. When you look at advanced stage lung cancer, comparing the 4-marker panel in red with the performance of ctDNA, well, there is some performance of the ctDNA. But when you look at early stages, the performance of ctDNA from the CancerSeek study had an AUC 0.61. Our marker panel had an AUC of 0.76. And when you add the ctDNA, the performance goes from 0.76 to 0.785, not much of an increased performance. Okay, well, is it still worth it adding ctDNA? Well, it may be a little bit questionable. We have looked at exosomes for liquid biopsy. There is some contribution there. I'm going to skip the slides. We have looked at autoantibodies. There's some contribution there. At the same time, with our 4-marker panel that we intended for screening, we asked, could this be used for indeterminate nodules? And so this is a paper that came out in JTO last year. And so when you look at the performance of the current way we evaluate nodules, and we are talking here about 6 millimeter or smaller nodules, compared to the performance of the marker panel, there's a huge increment in performance, very high sensitivity at 100% specificity in telling you whether this marker is cancer or not cancer. Now, we have worked with outside cohort, but we have launched our own cohorts at MD Anderson. One is the LEAP, Lung Cancer Early Detection Assessment and Prevention Cohort, which is based in the USA, primarily at MD Anderson, but UT Tyler, USC, and some regional centers. And we had other sites outside, 2 sites in Europe, in Paris and in Madrid, at MD Anderson Madrid, and a big contribution from China in 3 provinces. And so the Chinese component has contributed more than 6,000 subjects that we have blood data stored in at CRO in China, in Shanghai now, that we wish we could bring over. And at the other sites, we have some 3,000 subjects that have been accrued. And at the present time, outside of China, we have 63 lung cancers that were diagnosed and 36 others. And we feel that this cohort, the LEAP cohort, would be very valuable for doing ultimate validation of the marker panel, and perhaps we're taking it to the FDA. We have launched another cohort, the MERIT study cohort, mammography, early detection, risk assessment, imaging technologies, that we are very, very proud of at MD Anderson. We have some 18,000 women that come for mammography screening and MD Anderson. Why not develop a cohort with those women coming for screening? And so the idea, again, like with lung cancer, integrate imaging with subject characteristics and omics type of marker with an ultimate goal of overhauling screening for breast cancer. And we have accrued so far some 6,000 subjects. We have 12,667 mammogram, more than 10,000 blood draws. 89 have been diagnosed with breast cancer, 49 with other cancers. Those two cohorts would be great for not only lung and breast, but also for any pan-cancer applications. I see that I'm running out of time, so I got to skip a few of the slides at the end and basically acknowledge it takes a real village to be able to do this kind of work. And it's a long list of contributors. I don't want to skip anyone or point out to any one person. We have a lot of people in the lab that have contributed tremendously. I'm only listing here the lung cancer collaborators and overlooking all the work that's been done with collaborators on pancreas, ovarian, and breast, and other and colon and other cancers. And this has required a lot of effort, and we are appreciative of the support that we got from the EDRN at NCI, from NCI U01 projects, the DOD, the CPRIT, the Lyda Hill Foundation, and the MD Anderson Moonshot. So I skipped a few slides. I'm sorry about that, but I want to make sure I'm leaving time here for questions. And thank you very much. This is the finish line that we hope to reach someday. You see the cancer MD Anderson logo here. And thank you very much for being a part of this presentation.
Thank you very much, Dr. Hanashi. It was in fact very interesting and very helpful for all of us. And I start with the first question from Kati Panabier. Thank you, Kati, for being with us. She said, "Dear Sam, excellent talk. I feel that early detection of cancer will be feasible using, first, a combination of different circulating biomarkers, so-called liquid biopsy, and second, a higher blood volume that then used for the advanced cancer." So what is your opinion? You already hit some of these points.
In terms of blood volume, we think one has to be realistic. We go to the doctor for a checkup and we get our cholesterol checked. And if somebody is a smoker or for some reason they may be at risk for lung cancer, getting another tube of blood to be drawn for a lung cancer blood test should be okay, but requiring a gallon of blood or a liter of blood or 200 milliliters so that you could get adequate representation of a rare sort of molecule, I think this would be unrealistic. Now, there's another question about circulating tumor cells. So we were interested in circulating tumor cells. One of our colleagues in France, who was a very big advocate of circulating tumor cells, basically did a study involving a large number of centers in France, and for subjects who were COPD patients and wanting to do screening for those COPD patients, asking, would CTCs be good enough to catch lung cancer among the COPD patients? And the answer was a flat no. They were extremely disappointed that the CTCs were not useful at all in catching lung cancer early. And one day he picks up the phone and says, well, Sam, we have all those blood that were collected. Would you be interested in testing your markers? We said, well, sure, Paul, we would be happy to. And so we got all of those samples blinded that we were doing yet another validation study for the contribution of biomarkers for risk assessment and early detection among COPD patients.
Thank you very much. So in fact, you read, I think you read with your mind the second question was exactly this one. What about circulating tumor cells? Are they still relevant to the story of earlier detection?
Yeah, that's the question I just answered. Based on the validation study that was done in France for lung cancer, the circulating tumor cells did not contribute to catching lung cancer or assessing risk.
Thank you. Now I move forward to the next question by Shumei. So the 4-panel in lung cancer can be used for other tumor types?
Well, that's a very good question. We have a multitude of markers for the different cancer types that I listed. Some overlap, and some are at the present time appear to be unique to that cancer type. So there's some overlap in markers. For example, for pancreatic cancer, we have a list of 8 to 10 markers. Some of them are common to other cancers, but some appear to be unique. The same for breast, the same for colon, the same for lung. And so at the end, if we wanted to develop a pan-cancer type, then we would have to include markers that are common and markers that are unique to be able to detect cancer and to be able to determine the tissue of origin.
Thank you very much. Now to come back to a comment by Kathy. Kathy was saying CTCs were detected by a specific ISET technology. So this shows she feels that the question is still open about the CTCs and the detection. So this is coming with a new question for you, Sam. What about the technologies and the limitation of the technologies who are used?
This is sort of like a pervasive issue for any sort of marker. Now, if the marker doesn't perform, is it the fault of the marker, or is it the fault of the technology that you are using? So, or is it the fault of the design of the study or how the samples were collected, what have you? So one could say that one negative study doesn't really rule out the potential of a given marker or marker type, but it makes it a little bit more challenging to want to come up with a better test for the same marker. So all we can do is to go by the evidence out there. The people in France were high advocates, big advocates of CTCs, and that's why they did the validation study, but it didn't work out for them. That's all I can say. And so at our end, like in my lab, in my group, we have not pursued further circulating tumor cells.
Thank you very much. Another interesting question. So it's about extracellular vesicles that are negatively charged. So the question is, could you suggest if EVs in the natural form will get rejected by the body if employed for drug delivery vaccine development? It is essential to engineer the surface of the EV to make them neutral, to be accepted by the body.
Well, we have published on this, and I mentioned very briefly that we have done a lot of work looking for autoantibodies against different tumor types. In the studies that we did for pancreatic cancer, we pulled out from the blood those antigens that were associated with autoantibodies. And so there was a whole lot of antigens that we were able to pull out from pancreatic cancer plasma compared to control plasma. And when we did sort of an analysis of the data in terms of gene enrichment, it was clear that a lot of what we found were proteins associated with exosomes. And we did a lot of studies after that showed that actually exosomes in the blood are associated with an immune response. In our case, in the case of the work that we did, were associated with an autoantibody response. And we did further work that shows that, in fact, the circulating exosomes were hijacking the immune response. So instead of the immune response being directed against the tumor, now the immune response is being directed against the exosomes. So that made us think that it will be challenging to use exosomes for any kind of therapy because they are going to be associated with an immune response, and whatever their cargo is going to be sort of negated. Are there ways that you could overcome that concern? Well, yeah, of course, there are ways that you could do that, but we sort of raised a red flag a little bit at the time about what the exosomes are doing, given what they can do, given that they are hijacking the immune response.
Thank you. Now a couple of questions were connected. So first, how long do you think it will take till this type of early detection biomarkers will become a part of the physical exam for everyone, like preventive screening at your doctor's office?
Well, that's a good question. One could be optimistic and one could be pessimistic. It sort of reminds me of Francis Collins when the genome was sequenced and he predicted that by the year 2010, we will have a cure for every disease. And obviously this has not happened. I would like to think that this is going to be incremental. It's not like overnight we're going to have a test that will be effective for every which cancer. I think it would be implemented perhaps in an effective way gradually. And the approach that we're following is let's go after the common cancers and develop very good tests for the common cancers. Before we work on 50 cancers, and let's try to learn to walk before we run. And so that's what we're doing at the present time, wanting to come up with a good test for lung cancer, a good test for pancreatic cancer, a good test for breast cancer. Eventually, we feel that a combination, a multi-cancer panel could become very useful. And so this will be gradual, not an overnight thing that all of a sudden we have a very effective test for all sorts of cancers.
Thank you very much. And now one more question, because in fact, the talk was very interesting and also very practical. A practical question. What are, according to you, the barriers to get this liquid biopsy as part of a standard of care? Are FDA approval difficulties, insurance approval difficulties, and what are some examples if there are such pending approvals for lung or any type of cancer for the biomarker system?
Well, that's a great question. So now in the US in particular, then for a test to be widely used, there are a lot of barriers that need to be overcome. And FDA is not the only barrier. You have FDA, you have the US Preventive Services Task Force that has to recommend the test, and you have CMS that has to approve reimbursement for that test. We have all of those requirements in the US, but in Japan, they don't have those types of requirements. And so we are a little bit handicapped from that point of view. And for USPSTF, for CMS, and for FDA to approve, obviously, the test has to meet certain criteria, which is very important. But beyond that, there has to be a public sort of desire for this. And Congress has to be behind it. So right now, there's a lot of talk and a potential for Congress to come up with some legislation related to multi-cancer early detection, because quite a few stakeholders are pushing for it. And so when that becomes available, I'm not sure, but again, I'm being cautious here by stating that dealing with one cancer at a time has a much better prospect than FDA approving a 50 cancer lung detection test all at the same time. But I could be wrong.
Thank you very much. So with this one, I want to thank very much to Dr. Sam Hanash and to all of you. In fact, it's wonderful to see Passion for Science and Passion for Life in these very difficult times. And in fact, Sam, your presentation was wonderful because it brings us new ideas how to save lives. And with this, I want to tell to everyone, good luck with everything. Please, if you can, visit my LinkedIn page and widespread the very important message that it's there, because in fact we have to say a very strong no to war and a very strong yes to life and to science. And with this, I'm waiting you in 2 weeks from now for another wonderful presentation by Jason Hughes. Thank you again, Dr. Hanash, and thank you to all of you, and good luck with everything.